water into the cells without walls.
Yeah.
All right, so here we are. This is where we left off. So the next couple slides are just a review of what we talked about yesterday with that plant and animal cell picture in the various solutions. So tonicity, the ability of a solution to cause a cell to gain or lose water, depending upon concentrations of the water. We're right here, water balance.
Oh. What was the one before that? No. Water flows up. Thank you.
Keep forgetting to bring batteries. I think it's going to stop working. There we go. All right, so animals and other organisms without cell walls have osmotic problems, meaning that water is going to keep on going into them, in either or out of them in a hyper or hypotonic environment. So we saw that yesterday. Go back.
Yeah, there's. You forgot. Oh. Alright, never mind. I thought you skipped that.
It's all right. It's all right.
Alright.
All right. I had it up there, then I went back, and then I went back again.
Yeah, I
so I
didn't
kind
know-
of did it kind of fast because I was
Yeah.
there previously.
Yeah, I
so.
didn't know like
Okay.
if you forgot to put that on there or
Yeah.
something.
All right, we're good. We're good. Go ahead. You got it?
Yeah.
All right. All right. So when you have a cell with that, to maintain their internal environment, such organisms must have adaptations for osmoregulation or regulating osmosis or the control of water balance. So an example of adaptation that we've talked about before is the protist, the unicellular organism called paramecium. Paramecium is hypertonic to its pond water environment and has a contractile vacuole that acts as a pump. So before I even go on here, when it says the paramecium is a unicellular organism, is hypertonic to its pond water environment. So you should be able in your head to visualize what that means. So where the higher concentration of water is, if it's inside the cell versus outside the cell. So this is what we know. This is hypertonic compared to its pond water environment. What does that mean about the pond water versus the inside?
How's higher concentration of water outside?
There's a higher concentration of water outside. So whatever that concentration is, there's a higher concentration of the outside that makes this hypotonic compared to the inside. So therefore, this has the lower concentration of water, and therefore water is going to move into the cell. So you should be able to think through that whole process by this one sentence. All right, so that's something you would have to be able to do. Okay, so and to combat it, because it does not have a cell wall like plants. Remember, the plants counteract the pressure of the water, and therefore plant cells don't explode. But these guys could, but they have the contractile vacuole that I've talked about before in the cell chapter. Was one of the vacuoles, and this is an actual real picture of a paramecium. This is the vacuole filling with water, and then when it contracts, and what you can't see here is the water squirting out back into the environment. And so therefore, even though water is diffusing into this little guy, the contractile vacuole will make it so that he does not explode like that animal cell that we saw, that red blood cell that we saw in the picture yesterday. So that is one adaptation. I'm going to go back to this here where it says adaptations. Another example of an adaptation is our cells. In our cells, the adaptation for our body is not, we don't have contractile vacuoles in our cells, but our cells are bathed in what? What kind of solution? Isotonic solution. So if you're bathed in an isotonic solution, that combats the whole, your cells exploding and so on. All right? And so some multicellular organisms like us, that's one of the ways in which we help to control water balance. Okay, so then let's look at the cells with walls. Well, like I said, we've already talked about this. So cell walls help maintain water balance. plant cell in a hypotonic solution swells until the wall opposes the uptake of the water, and we say that the cell is now turgid or firm. And if the plant and its surroundings are isotonic, then there's no net movement of water into the cell, and the cell becomes more limp, and the plant may wilt. So that's why we said yesterday that the ideal solution for plants is a hypotonic solution. In a hypertonic solution, the plant cells are going to lose water, and eventually that membrane pulls away from the cell wall. Usually a lethal effect. Lethal means deadly, right? And that's called plasmolysis, and so that's the picture we saw yesterday. And so, hypotonic is not good for plants nor animal cells. And so this is the exact, close to the exact same picture. It's the same information in it. I just wanted you to think through the information yourself yesterday. So this is the exact. It's kind of flipped around a little bit, like hyper was over here in your picture and hypo was over here and so on, but it's the same information. They just added a few vocabulary terms here underneath. So notice here when the cell explodes, another way to say that is it's lysed. It means to poke, split or cut or poke holes in. And so this is a normal cell for animal cells, so that's the ideal situation, and this is shriveled up. When we look at the plant cells in a hypotonic solution, this is the normal condition or ideal condition for plant cells to be hypotonic, and it's turgid, meaning very rigid and stands up very straight. Flaccid means that it is limp and the water the plant can wilt, and this is the plasmolysed cell. Remember the cell membrane here shrinking away from the membrane because of the volume of water decreasing inside. All right, and that in both of these cases for plant and animal cells will cause the cell to die. All right, so as we talk about things moving into and out of the cell and why they do, from high to low concentration, and we looked at the different examples of diffusion and osmosis. Remember that some things can get through the main part of the membrane, which is the lipid bilayer. Do you guys remember what kinds of things can get through the lipid bilayer? It has to do with, there's two things that determine polarity and something else. So what? Non-polar things can go through. So hydrophobic molecules, and that's because remember that the lipid bilayer, the main part of the thickness of the lipid bilayer is this non-polar region here, and so therefore non-polar things can move through that lipid bilayer. So non-polar or hydrophobic things can get through the lipid bilayer. What's the second type of molecule that can get through the lipid bilayer? Sam?
Small, uncharged, polar.
That's right. So size makes a difference. So the small molecules that are polar can get through here as well. So small polar. That leads other things that cannot get through the lipid bilayer, either not getting into or out of the cell, or they need help with that. And the help comes from proteins. So there are proteins also embedded in our lipid bilayer that help other things go through. And those would be large polar molecules and ions. And one exception of a small polar molecule is water. I had told you that water, there's actually little proteins called aquaporins that water goes through. So let's talk about these transport proteins that allow, get near the end of the hour, aren't we? All right. So we'll transport proteins like that. All right. Facilitate passage of substances across the membrane that don't move by simple diffusion. Write this, simple diffusion, what that means is through the lipid bilayer. So if you can't get through the lipid bilayer, it has to go through a protein. And this is where we'll stop. We'll pick this up tomorrow.